Hello, thanks for being interested in our research. We are a group from Auburn University and VCOM-Auburn, and we are happy to present our work on urine sensors in this IEEE Sensors Conference 2021. In this work, we have developed a sensor platform that is capable of detecting bacteria for urinary tract infections. Urinary tract infections, or UTI, is one of the most common urological diseases in the United States and costs 10 millions of healthcare visits and billions of dollars each year. It is meaningful to have something that can detect this kind of infections early and monitor it. The device we developed is a magnetostrictive particle sensor, which is a wireless sensing system that is low cost and can generate the result in real time. Compared with the current method used in the clinic, such as the spread culture method or PCR, our method is easier, safer, and cheaper to operate. There is no cultivation process needed, and once the sensors are made, they are label-free. The principle of the system is based on the magnetostrictive effect, that some metal alloys can change their dimensions and vibrate in changing magnet field. Such vibrations would have a defined resonance frequency. This frequency can change along with the mass change of the metal piece. If we link the biorecognition unit to such metal piece, they could become sensors. The change in the resonance frequency becomes the signal we can acquire by a pickup coil to monitor the capture of the target cells. In this experiment, we use the E. coli as an example, as the E. coli accounts for about 85% of the total UTI occurrence. We have modified the sensor with antibodies and linked them to the gold coating surface, and later proved that the corrosion of the urine did not have a significant impact to our sensor system. The signal acquired is robust and stable. We create a closed system that allows the urine which contains the E. coli to pass through the sensor. Our preliminary data has shown that at 10 to the 6 CFU per milliliter concentration, the bonding process of the bacteria is reflected clearly by the frequency change. Only 5 milliliter samples were used in each experiment, and within the first 60 minutes, the frequency has a stable trend of decreasing. The peak is moving from lighter color to a darker color with time as shown in the picture. We now know that this concentration could be further reduced, and 10 to the 6 is not the detection limit of our system. In addition, when we fit the frequency signal data and analyze the change of the frequency shift, the result becomes more obvious that compared to the control group, the urine sample with E. coli induced a much higher frequency shift and higher slope. And such frequency shift become quite noticeable in as little as 20 minutes. Therefore, we can conclude that our sensor platform could be used effectively for urine sample, and did take the E. coli, which is #1 UTI cause, as the example for demonstration. The antibody used in this experiment could be easily changed for targeting other pathogens. So our system really provides an approach for diagnosing UTI and detecting other pathogens in urine. Thank you.